So thank you all for coming out here to hear a little bit about this field of 3D printing. I call my title Shaping Medicine because I'm going to talk about ways in which we use 3D printing to define both the shape and the structure of biomaterials that we can then use to implant into patients to hopefully treat them for a wide range of medical applications. Some of this is already a reality. We're already putting implants into patients that have been 3D printed to match things like the shape of their defects. And then we can think a little bit into the future about where we can do things like organ 3D printing. So we're not quite there yet, but we hope to be there soon. And I have to start by thanking my laboratory. So this is us just a few months ago up at Lake Isabel. We had done a nice hike up there. Many of them are here today, and they don't often get to hear me talk except for classes. So it's a lot of fun. And all the great images and movies and stuff that I'll show you today, they're the ones that actually did all of that. All right, so is this science fiction or is this reality? So I was trying to think of ways in which we see 3D printing surrounding us in media. These are some images from a TV show called Westworld that maybe you remember. It came out about 10 years ago. This was right when we were getting excited about 3D printing. So it was fun to see these shows where they're talking about actually 3D printing whole organisms. So I took some snapshots of just the introduction to this. And what I liked about this is it's actually showing that they're 3D printing these objects, these tissues, and all this is just a little printer, much like this one that I'll introduce you to in a little bit. And I also like that they did this buffalo. I thought that was very appropriate for CU Boulder. And it's going to stop here, and what you're going to see is that all this is a simple 3D printer that's extruding this little bit of material, depositing it to build up this 3D structure. So maybe that's got cells in it, maybe it's just a material, but that's the way we can think about advancing this. So I'll give examples again where this is a bit of a reality. We'll talk about things that are maybe just a few years away, and then we'll talk about things that might sound like science fiction right now, but will be reality in the future. Okay, so the outline of my talk, I suspect there's a wide range of backgrounds on 3D printing in the audience, so I'm going to tell you a little bit about what this is and how it works, give you a few examples of where it's used. I'm going to move to a story that I like. Sorry, this is a little bit graphic, this image, but this is an idea of a 3D printed patch that can be placed on a tissue, in this case in a heart, and these patches can do things like seal leaks in tissues. They can deliver signals to help those tissues repair, so they can carry cells and drugs and things. So I'll tell you what we've been doing with this patch system. And then I'll spend the second half of my presentation talking about living materials, so actually 3D printing with cells to make little tiny tissues. We're pretty good at that. And we can use that for things like screening drugs. So maybe that's a way in which we can better replicate how a drug might behave in a human. And then I'll move the story a little bit with some tissue engineering, and then I'll finish with some thoughts on full organ 3D printing. So we would love to do something like this, but right now that's just a cartoon. But hopefully we can do that in the future. So I'll show you kind of where we've been going with that. Okay, so what is 3D printing? So by way of hands, can I ask who's actually done 3D printing before? That's a pretty good group, about a third of the room, maybe even closer to a half. I asked this to my class that I teach, and I'd say, 10 years ago, I'd ask, has anyone done 3D printing? And maybe a few people had. I'm teaching a class right now on tissue engineering, and I asked, has anyone done 3D printing? Everyone's done 3D printing. So this is something that's integrating itself into education, into outreach. And so I think it's getting people excited about science and technology. So does anyone have a 3D printer at home? Couple people. So maybe I'll ask, what types of things have you 3D printed before?
Stuff for my kids, and also I fixed my house.
You fix your house. So this is not a random person in the audience. This is my good friend Kat, who I've known for 25 years. And when I was coming back to CU Boulder, I visited Kat, and she kept going out to her garage, and I was like, what are you doing out in your garage? And she's like, I'm 3D printing this part for my bike. And she'd print a part, try it, it wouldn't fit, she'd redesign it, print a part, and do that. So 3D printing is going to find its way into many of our households, I think, for day-to-day use. Okay, so let's step back. How do we make and manufacture material? So if I look at things around me that are made out of plastics, there's different technologies that I can use. So one of them is called injection molding. And this just means that I make a solid mold that has some cavity in it that's meant to be the shape of the part that you want to make. And then you just inject this liquid solution. Usually it's a heated up plastic material. It fills that void, then it cools down, and you open it up and you take that part out. This is fantastic for many applications if you want to make the exact same thing over and over and over. But where it fails, if I want to redesign this, I have to make a whole new mold. So this can cost a lot of money. And so that doesn't always work for things where you want to redesign it all the time. Another method you might think about is this subtractive manufacturing or machining. So imagine you just have a block of material and you just carve away at that material to leave behind this three-dimensional shape. So that's great because I can design any shape that I want to carve that into, but it has some challenges if I want to put in a three-dimensional structure within that material. So it's very good to make things like statues. So if you want to take marble, this is Michelangelo's David, you can carve that away. But again, that's just a solid piece of material. So you kind of see where I'm headed with this. 3D printing solves these problems. So 3D printing is what we call additive manufacturing. So as this movie is showing, it's just adding material where we tell it to build up this three-dimensional structure. So printing things like this open porous structure or something like this, that would be hard to do using these injection molding techniques or these carving approaches. And so that's why we like 3D printing. It gives us the ability to design any system that we want and then go about 3D printing it. Okay, so what are the steps to doing this? So this is kind of a simplified process. So first of all, we need to think about what we want to print. And then we just make a computer file that tells us what that shape is. And there's lots of ways to come up with this file. So you can go, the National Institute of Health has this repository where I can download all of these files of different types of shapes. So I just typed in heart, and this is what popped up. So you can go and find this online. You can also use things like imaging. So if you have a patient and you want to see what the shape of their defect is, you might use something like MRI, and that'll tell you exactly the shape that you want to 3D print. And lastly, you can just step back and you can design anything you want. So my lab's very good at this. I've never done this before, but they're good at really putting together these files to tell us what to print. And so we use 3D printing for so many different things these days in the laboratory. So one way to look at this is then, once you have that file, what you need to do next is just slice it. And that just means I'm going to cut it into tiny pieces, tiny layers, because 3D printing is usually a layer-by-layer approach. And that's going to tell the 3D printer where to deposit that material. So it's kind of a simple idea in concept, but it's kind of complex depending on what type of 3D printing you use. And then you just print it. So we've told the printer where to deposit that material to build up that 3D object. And then you're left with that final object. So this is an image that's often used to teach about 3D printing. So they often use this Yoda structure. And I think it's because we're all familiar with what that looks like. So we're really good at telling, did we print something well? And it has all these kind of unique features, these ears and the contours to the face. So again, this is the file that tells us what do we want that shape to be. And then we just go through those different steps, and then we end up with a 3D printed structure based on that design. So I'm trying to keep it as simple as possible, but just so you understand that it's a layer-by-layer additive manufacturing process. So one of the most common ways that we 3D print things is shown here. This is a technique called fused deposition modeling, and this is about 35 years old. And probably most of you who have done 3D printing, this is what you've actually used for 3D printing. All this is you take this plastic material, it's just a thin filament, and then that feeds through the printer head. And as it does that, it just heats up that plastic. So again, that turns it into a bit of a liquid. And then when it comes out of that printer head, then it just flows, and then it's deposited where that printer told it to go. And then because it can cool down, it just solidifies. So kind of a simple idea, again, just uses a little bit of that melting of that plastic to then deposit and to control where that material is deposited. So again, this is the type of 3D printing that is used. So this is Chris Hiley. Chris Hiley was the first person in my lab that started to do 3D printing. So this is over a decade ago. Chris is now a faculty member at the University of Virginia. Chris came to me about maybe 3D printing. He nicely saw that some of the materials we've been working with might be really useful in 3D printing. So we thought about buying a 3D printer, and we looked up how much those cost. And at the time, it was about $100,000 to just buy a 3D printer with really good functionality, but it wasn't so cheap. And so Chris went back and he looked at what was freely available for different types of software and hardware, and he essentially just built his own 3D printer. So he built this for about $500 and has all the functionality of what we could have bought from that company. And he even really designed his own kind of printing apparatus up here. And this is that printer at work. So this is printing out this kind of gel-like substance and it's just extruding it. So it's just like a syringe, but we're controlling where it's depositing. So a simple printer, and we use this for many years. In fact, we often like these self-built printers because we can really control what we do with it. So this became the workhorse for the laboratory for a lot of the work we did in bioprinting and 3D printing. I really like this idea of open source. So there's been a lot of examples now in the literature. So one of my colleagues at Stanford developed this printer called the Printess. And they basically teach people how to build their own printer. As you can see, this is a printer right here that we self-built. All you have to do is use things like laser cutting to make these plastics in different shapes. You can use 3D printing of certain parts. You buy some motors, and then you just put them together into this 3D printer. So I teach a class for graduate students on biofabrication, and the first few labs we go through and we just build this printer. So this is intended to take a few hours to build. So the students do it over a few class periods. And again, this is the printer that we can build. So it basically has 2 printer heads, so just 2 syringes, and each of those can deposit different material. So you're welcome to come look at this one after the talk if you're interested. And thanks to Arkadeep, who's here, because he really ran this laboratory to build those printers. Okay, so what can we do with 3D printing? So here's some examples. So these are custom metal implants that have been 3D printed to fit a hip joint or to fit some sort of defect in the skull. So those have been 3D printed using different technology. And in white here, these are 3D printed kind of replicated structures of those patients so that you can see whether or not that object actually fits into that defect. So you can use printing both for the implant and to kind of understand that material. And here's some example from dentistry. This is an area where 3D printing is making a big impact. So printing things like crowns, or in craniofacial reconstruction, printing ceramics so that they can be transplanted. These are happening right now. So these are types of therapies and types of things that you can actually go and get right now. Other types of materials, so there's really diverse properties. Here's just an example of a hard plastic that's used for an external cast. Here's some examples of things like a heart valve, or this is an ear that's being squished. So this is from our work in our laboratory. So each of these needs really different material properties. They're all plastics, but they're designed so some are stretchy, some are really hard. So depending on the application, we can now print a wide range of materials. And then the last material that I'll talk about are called hydrogels or gels. And so this is something like a contact lens or gelatin. So these are really soft materials. They have a lot of water in them. So imagine your contact lens. If you put it on your counter, it would dry out over time. So they're made out of solids that really like water, so they swell. And so something like gelatin, so if you take Jell-O, you melt it down, heat it up, and then when you cool it down, that just is this gelation process where it's making this solid material. So here's just an example of 3D printing these types of really soft materials. So in this case, just like in this printer, you're just extruding that material and controlling where you're depositing it in 3D space. So my last kind of intro slide here about 3D printing, I want to make a point that we can do 3D printing on the really small scale or really large scale. So here's an example from a colleague of mine in Vienna, Alexander Ozakonov. What they did is they used a light-based printing method. So this is just a normal pencil, and this little box up here is a magnification or kind of outlines what this magnification is. So they use this technology to make this little castle on the tip of a pencil. And it's just by controlling this light exposure to this material that they could do it. So don't think 3D printing has to be big. It can be really, really tiny. So now these are features that are maybe similar to the size of your hair strand or a cell. We can also make these really big. So this is an example. I was at a conference a few years ago, and they showed this example from an artist that was using 3D printing to build a house. So this is the printer head, and all it's doing is extruding cement as it's building up into this 3D structure. So again, this is a really large scale, meters-based scale that you can do to make these larger objects. So this is what is exciting about 3D printing. It means a lot of different things, but it's really useful. Okay, so I'm going to tell you about one project in the laboratory, and this is to make adhesive patches that can be useful in medicine. So some examples where this might be useful is this is an example of intestinal tissue where maybe you've removed some tissue due to something like a tumor. So rather than just suturing, you could think of a patch that would go in and basically seal that tissue up. So here they call this a duct tape-like patch. On the right here is a heart. That heart also has this patch on it. So this might be after a heart attack. This tissue is damaged. So I can put a patch on there and try to maybe heal that tissue by delivering things like drugs and cells. So lots of reasons to use these different patches. The challenges, though, are designing a material that can behave as we want it to in the body. So imagine that you have a Band-Aid that you put on your skin. Try doing that when you're kind of submerged in a swimming pool or underwater. It's really hard to make that sticky. So that's a big challenge. We have to think about this hydrated, moist environment and how we can then make these things adhesive. And then the other thing is that tissues move and stretch. Your heart is continuing to beat over and over and over. So if I put something on it that's really too solid and that beats, it's just going to fall off. So these are our design criteria. So these are useful for lots of applications. Maybe you've seen this video of this infomercial called Flex Tape. So people bring this up all the time when I show these examples. So I took the audio off because this guy yells a lot. So this is Phil Swift. Phil has a lot of leaking containers in his room, and he just slaps this patch on. That's exactly what we want to do. Phil's very excited about what he's done. But we need to do this now with materials that can be received in the body and using mechanisms that are very different than how this attaches to plastic. So bring it back to kind of the biomedical standing. So what are our design criteria? So we're engineers, so we start to think about what do we need to design into these systems. So the first thing is that we want hydrogels that are really tough. So toughness means that I can apply a load to that material and it can deform and stretch and not break down. This material that I'll show you is not tough. So you can see you pull on it and it just fractures. So that material, we would call that brittle. That doesn't make a very good adhesive patch because it can't move around. But here's a material that's really tough. So we just made these yesterday. So thank you to Jonathan and Matt for putting these together. And you can see this is a material that I can stretch and it deforms. So that's the type of thing that we want to see on like a heart tissue because then when it moves around, it's going to stay on there. So that's first criteria number one. And then of course we need it to be adhesive. I'm not going to go through the chemistry that we use, but there's certain tricks we can introduce to do that. And then we want to 3D print it for large, to really control the shape and size of these. Okay, so now we have to 3D print it. Thank you to Elise who gave me this video. We now go to a completely different 3D printing. So hopefully I don't confuse you, but again, this is a layer by layer 3D printing. This yellow liquid is a liquid that when I expose it to light, it solidifies. So it just takes a little bit of light. And what this printer is doing is each layer, it's putting a thin layer of that fluid, that yellow liquid, and then it's exposing it from the bottom, just like our projector is, to some pattern of light. So each layer then we can define what that pattern is. And if you do that over and over and you change that pattern, now I've defined this three-dimensional shape. So this is called digital light projection, and it's a layer by layer approach, and it's all about light exposure. And light is great because you can really control where this material then changes. And so that's the object that's been 3D printed, and so you can see it just sitting right there. So now that's the type of patch or something that we can print. So we were able to do this. We were able to make tough materials into 3D printed objects. Here's just a few things that we printed that show we can make these nice structures. And here's Matt, who's 3D printed this material and just showing that we can stretch it. So again, it has that toughness or that ability to deform that we were looking for. Here's just a few images of movies. These are all 3D printed objects. So imagine if you could do this with your contact lens, it would just tear or rip. And so these tough materials then can withstand all of these types of loading. And then we do this very technical test where we just run it over with a bike. And you can see, again, you can put that load on it, and the material returns completely back to its original state or that original shape. Okay. So this is an example of the material shown here, and this is a small piece of tissue, and we've added some chemistry that then links those together. So that's the mechanism of which it'll stick to that tissue. And on the right here, again, is just a heart. So this image shows us putting a patch on the heart. So we just place it in contact for tens of seconds, and then on the right, just showing that it's sticky. It's stuck to that tissue, and this lattice structure makes for a really nice mold because it kind of stretches as that heart continues to beat. So this is what we were excited about. So we were so excited about this, we started a company. And as Roy had said, BioFrontiers has been a great community for entrepreneurship. And CU Boulder has been fantastic. So we just pitched a few weeks ago to a program called Lab Venture Challenge and raised a little money for this company. And this is something that Matt and I started. And this is our tagline. So bioadhesives that seal where sutures fail. So we're looking for applications that are really hard to use sutures in. Maybe it's hard because of the properties of that tissue. So in a lot of spinal surgeries, so this is an example of the vertebrae, and in here is your spinal cord, and it's covered in a thin layer of tissue called the dural tissue. And in about 10% of surgeries that go into the spine, you're going to tear that tissue. So either through the procedure, you might cut it. And when you do that, you leak out this cerebrospinal fluid, which can cause a lot of problems for patients. So things like chronic headaches. And it can also cost a lot of time, a lot of money as well. So if we could develop a patch, such as DuraPatch, we can hopefully then seal that. So a one-step patch that just goes on, much like that video. Matt doesn't really do that with those patches, but this is what the patch looks like. So it's a 3D printed patch that has some chemistry between the patch, and then this is that dura mater tissue. So this is a similar video to that one that you had seen. We put some dural tissue on this reservoir, and you see if you puncture it, it just leaks out. And then Matt just comes in with this patch, much more gentle, I think, than you saw earlier. But just within seconds, it sticks to that tissue. And so that's really exciting. This is what we wanted to do. So this is a company that we started. It's purely Colorado-based company. So we have some collaborators at the Anschutz Medical Campus. They're collaborators up at the vet school at Colorado State University as well. So we're having a lot of fun kind of exploring this company and where it can go. Okay, so I'm about halfway through my presentation. I'm now going to completely switch to now more living materials. So I'm going to get into living tissue structures and organs. So moving from some of those devices that are 3D printed and thinking about actually engineering tissues and cells. So I think the motivation for this is fairly obvious. This is just some examples of the lists of patients that are looking for organ transplants and don't have enough organs available. So for things like kidneys, livers, hearts, the pancreas. And this is an example over here of how long are these patients waiting on these lists, and it's up to 4 or 5 years. So this is a problem because these patients are quite sick. So if they're on these long lists for their organs for transplantation, this can be a problem. So we want to solve that problem. So to do this, we have to think about what are tissues and organs. So they're made up of things such as cells. These cells are surrounded by a matrix. And there's many tissues that have different cell populations, and how they interact with each other becomes really important in the function of that tissue. This shows the liver, which I'll come back to in a little bit. And you can see it's fairly complex as far as what actually goes into that liver. And it's that complexity that makes it work and makes it function. So to get at this, we're going to look at this field of tissue engineering. So Christy Anseth gave a nice talk last year in this program, if you were able to catch that. And she really went into some of the advances in tissue engineering, where you can combine three-dimensional materials to act as a framework for tissues. You can combine cells and then maybe other signaling factors that can tell those cells what to do. So all of this combined becomes this tissue engineering approach. I'm going to give some examples, but we're going to focus on how 3D printing is used then to define these different shapes and structures of these materials. Okay, so where have we done really good as a field? So here's 2 examples that have been very successful. So tissue engineering of a bladder. These have been transplanted into patients. And then skin has been a tissue that has really been many products out there. And what's similar to these 2 tissues is that they're fairly simple. They're just a couple cell layers thick. And so we can grow these really quite nicely in the laboratory. If you think of other tissues, though, this is an example of the liver. So the liver has this really complex vessel structure. So this is where the blood feeds the liver. And you can see it branches into all of these different structures and patterns. That's really important. I actually talked about this earlier today in my tissue engineering class, how important it is to design this because cells can't live unless they're getting oxygen and blood from these types of vessels. Then if you look at the human liver, look how big this is. So it's liters in volume, large amount of blood vessels, and 100 billion liver cells. And that's not one cell type, that's multiple cell types that their organization becomes really important. So these are the challenges for organ tissue engineering. Complex vessel structures, really high cell densities, and then these large volumes. So I'm going to ignore that for a minute. I'm going to start talking about small tissues that we've been pretty good at engineering. So I thought I'd tell a quick story about how we can make small tissues and use them for things like drug screening. So the motivation here is that through drug development, we start with basic research in the laboratory. We move it into things like animal models that hopefully tell us whether these drugs are working. And then we move it into people. So we go through these phase 1, phase 2, and phase 3 clinical trials. And what this is meant to show is that all these drugs that we start with kind of narrow down, and maybe we're left with one drug that's actually approved by the Food and Drug Administration. So it's great that we have successes of these drugs being approved, but all of these that go into the clinical trial are lost or missed opportunities. And so what a lot of us think is that over here, we need to be better about how we're actually analyzing these drugs to better understand how they might behave in patients. So I think how we look at drugs and how they behave needs to be updated a little bit. So how do we test drugs? So a lot of it's just cell culture. So we just grow some cells in the laboratory, such as in this image, we put drugs on it, and we see how those cells do. So that's good because we can do it in high throughput, meaning we can do lots of experiments very quickly. But its relevance to the actual physiology of that disease is really low. So it's not replicating how those cells should behave or will behave in a human. If we look at animal models, such as rats, and maybe we go to large animal models like pigs, these have a better relevance as far as the physiology. But in most of these cases, we induce a disease or introduce a disease that doesn't match how a human actually develops that disease. So they often don't then replicate that, and we're not using human cells. So what's really exciting in this field is that we can make small engineered tissue models, and we can make these out of human cells. We can introduce things like fluid flow to mimic maybe how those blood vessels interact with those cells. And this becomes something that can hopefully mimic better how a tissue in a human will behave compared to some of these other options. And what's great is the FDA, so the Food and Drug Administration, has opened up this as an option, so just about 3 years ago, as an alternative to animal models. So we don't always need to use animal models now to move these types of therapeutics into patients. We can think about these types of platforms. Okay, so this goes back to work that we had started with Leslie Leinwan when I moved my lab here about 4 years ago. And I think it goes back to my rotation a little bit in her laboratory of trying to grow adult cardiac cells in the laboratory. I'd argue we still can't do that very well. These cells just don't live outside, so I think I got put on a really bad project by Leslie. If you look at this heart, you can see how the heart can continue to pump and function just for short periods of time after it's removed from the body. But the problem is these cells don't like this environment and they won't stay alive. So we need to rethink then, how are we growing and culturing cells in the laboratory? So in this heart tissue model, we have to think about adding these heart cells. These are these contractile cells that make the heart pump. We can introduce some alignment or organization to these cells, similar to what they see in a tissue. And then we want ways to be able to measure how these heart cells do. So we assembled a team, a great team of graduate students, Abhishek and Miranda, and postdoc Tommy, who came together to think about ways to take cells and make these little models out of it. So what we did, we're not actually printing the cells, we're printing a mold that becomes a reservoir for those cells. This is what it looks like, so these are hydrogels. This is what it looks like from the top. This is what it looks like from the side, so you can even see that layer by layer because we printed them. And this is a reservoir that we're gonna then fill with these cells and a little bit of other molecules. So in this case, we're using collagen, because collagen is a protein that's found within cardiac tissue. And we're using these cardiac cells down here that come from stem cells. So we fill up that little well, and it's got these little pillars here. And over time, what happens is these cells start to change their environment. So they're pulling in this collagen, and they're forming these little tissues here. And it's guided in this direction because of these pillars. So that's telling it to organize into that kind of structure. We can do this with lots of little ones. So this is nice because we can make, each of these are just a few millimeters. So we can, in one kind of cell culture dish, we can put tons of these so we can look at all these different drugs. And when you look at these, they look like small pieces of heart tissue. So you can see these are contracting up top. They're kind of pulling on those posts that I showed you. Down here, this is measuring calcium. It's showing us that they're propagating some electrical activity. And if we look at the tissues, they look very similar to that of an adult heart tissue. So this is what we can then use for things like drug screening. And I didn't want to spend too much time on this, but I just wanted to show that there's a lot of things we can do with such models. So with Leslie's lab, we're interested in genetic mutations with different types of cardiac disease, like this hypertrophic cardiomyopathy, where there's this overgrowth or thickening of the heart muscle. And then we're really excited in my lab thinking about heart attacks and how do we treat patients after that. So we can introduce different types of cell populations, basically putting a scar into this. So thank you to Mackenzie who contributed these nice videos. So we're just starting to think about the ways that we can actually use these to screen drugs and investigate biology. Okay, so that's really small tissues that were guided through 3D printing. So let's talk a little bit about bioprinting. So this is the case where we're actually putting cells then, and we're printing cells using different types of materials. So the plan here then is you design then what you want to print, much like we did with Yoda and some of the other examples. But now the ink is consisting of one of these gels. It can contain the cells. So the cells are actually going to go through this printing process. And we put it in a printer like this one, and we can do this all really sterilely so that these cells stay alive. And then we fabricate a three-dimensional construct that looks like a tissue, and we can grow this then in the laboratory or we can transplant it. So I'll give you just one example of where this could be useful. So this is your knee joint, and the knee joint has this thin layer of cartilage on it. So cartilage is this white tissue. This is very healthy, this tissue, because you can see that nice shiny white cartilage. And then that covers the bone. So that just helps cushion our joints as we're moving. For many patients that develop things like osteoarthritis, it breaks down that tissue. So this might come from some sort of injury to that patient. Just over time, our joints change a little bit. This will be really painful for that patient. So over time, this will develop, patient will develop pain. So in many of these cases, then, this leads to a total joint replacement in almost 1 million individuals in the United States each year. So taking this healthy tissue, and now these are pieces of metal that are placed in your joint. It's actually quite successful, and it reduces pain very quickly. But we'd like to look at alternatives where we can actually put cells and tissue as a replacement rather than metals. So here's just an example of a video from a colleague of mine in the Netherlands. They were interested in printing, 3D printing then, cartilage on top of bone. And cartilage has different cell compositions at different areas in that tissue. And the idea is this small defect, you would go in and you would image what the shape and size of that defect is, how thick that defect is, and that's going to guide then how you 3D print this. So they then 3D print in this cartoon this structural material that's going to give it mechanical properties. And then each of these are inks that contain different types of cells. So this is the bone one. So it contains a certain type of cell that'll form bone tissue. It also contains things like growth factors that tell these cells what to do. And you just 3D print these. And it's going to build it up layer by layer. So again, just a really nice layered approach here, where it's the bone layer, the deep cartilage layer, the middle cartilage layer, and then this top cartilage layer. And then you would just take this and you'd either implant it into the patient or you would culture it in the laboratory. So this would be useful for very small defects in your cartilage. As another example, so instead of metal, maybe I want to put in a larger implant that would replace that metal. So this is a design then where this green color is the cartilage and this gray color is the bone. And so you can 3D print that. So this is just going to print it layer by layer, building it up, where spatially you can print a material that becomes cartilage and a material that becomes bone. And so it does this and replicates it over and over. It provides some other types of materials or plastics in there that give it some structure. But this is a living material that we could then culture in the laboratory or even think about transplanting. So these are a little bit more on the horizon, these large types of tissue defects, because there's so much function that they have to carry into that patient. But these are examples where you actually 3D print living cells. Okay, so I want to end with my last section thinking a little bit about whole organ 3D printing. So I told you I'd come back to this. So this is the idea, right, is that you're actually 3D printing tissues like a heart or a liver. If we could do this, this would be amazing for patient treatments. Okay, what are the alternatives? So maybe you've seen some stories about genetic engineering of pigs that can develop then organs that can be used for transplantation. This one was just from earlier or last week. This story had come out in CNN. So an alternative to what I'm going to tell you about is this idea that you would grow these in an animal and then transplant them. And the challenge here is then trying to engineer these animals so that once I transplant this, it's not rejected from that human patient. So this is a huge challenge. It's great that it's being investigated. And then in the end, this will never be human cells. This is meant to be an alternative for all those patients who don't have organs for transplantation. So I think it's important to think about the other alternatives out there. What 3D printing gives us, if we can address all of these challenges, is the ability to think about actually putting human cells and human tissues back into these patients. So I just wanted to remind you of some of the challenges that I mentioned. And I'm going to spend just a few slides talking about some ways that we're trying to address those challenges. So again, really complex vessels, high cell densities, these large volumes of tissues. Okay, so to do this, we can think about new ways to print materials. So this is back to Chris Hiley, who I mentioned earlier. This is a technology that we started, this video is not fantastic, started thinking about a number of years ago to make blood vessels. So it's shown here on the right side, and then it's also in this movie. And basically what it is there's a hydrogel here, so another gel-like material. When I put my needle into it, that material just yields, so it receives that needle, and then I move it around, and at the exact same time, I'm putting down an ink. So in this case, the ink is another gel, which is shown here in pink, and in these images, it's shown here in blue. So again, it's just printing those structures that I told you were really complex. And this is really nice. We've been using this technology for a number of years. Another postdoc came along, Kwang-Hoon Song, and he started to actually make blood vessels out of these. So in this case, this is the gel that we printed that little spiral into. But the gel that we printed into, then we can solidify it. And then we can take that ink that we had 3D printed in there and just wash that out. So basically, we're just making a channel. And so you can see when I put a dyed solution here, it's just going to flow through that channel. So it's just filling up that void space. So again, a blood vessel is just this open channel that blood flows through. So we're trying to make those. And then he took it a few steps further. He took blood vessel cells that are known to coat blood vessels, and he would seed those in there. So they're now coating or lining then these channels. So this looks a lot like a blood vessel. And then he did one more thing that was really interesting. He took these vessels, so this is another one of these open vessels, and he introduced different signals, so these start to then sprout small, tiny vessels. So these would replicate some of your larger vessels, whereas these would replicate things like capillaries, so these small vessels that fill up your tissues. And just like I told my students today, this is what's going to fill up a tissue to be able to feed it and keep that tissue alive. So for large organs, we need this type of technology. The other technology that I'll mention is this idea of going from just cells in suspension to really high cell densities. And to do that, rather than looking at single cells, we're making these little clusters. So this is about 1,000 cells. We just put it in these little wells, and over about 24 hours, they like to just interact with each other. So they stick to each other and form these balls of cells. So we can make a lot of these, and we can start to think of this is now the building block of a tissue because it has that high cell density. So this is some projects by Andy Daly, a previous postdoc in the lab. So here's an example where we use 3D printing then to really pattern where these aggregates go. Again, I'm thinking about ways to pattern up different types of cell populations. He just uses a little vacuum. He kind of connects to this cell aggregate, and then he just drives it through a gel. So it's just another way you can 3D print something. So then he's just going to drop it off. So then you can pattern in all these different aggregates. Again, imagine different types of cells that then we pattern into a three-dimensional structure. And I just wanted to show, this is an example from a paper from a few years ago where we have these different cell populations labeled either green or red. And he basically just patterned them through this 3D printing approach. The last thing I'll mention is rather than single aggregates, we need to make these large volumes of tissues. So can we take a lot of them and put them together? So each of these is a cluster of like 1,000 cells. So we can put these and develop them in the laboratory, make a lot of them. And this is work by Meg Cook and Nico DiCaprio in the lab. So now rather than just single cells like Andy was doing, or single aggregates, we're now just printing large volumes of these aggregates into these tissues. So this is just becoming a circle of that tissue. So we print it and deposit it, and then they fuse together into a solid that we can then remove. So again, I just want to highlight some of the technologies that are making that idea of organ printing possible. All right, so I'll end here again. So science fiction or reality. So I've shown you some examples of we're already using 3D printing in humans and patients through implants. Other examples, so some of these disease models, pharmaceutical companies are already using this. They're screening drugs already to try to understand better how they can behave. A little bit in the future, some of maybe the tissue engineering, and then a little bit science fiction, but we hope reality soon would be this idea of full-scale organ bioprinting. And then I'll just end with my lab again, and thank you all for your attention.